Integrated disease management in sugarcane. Sugarcane crop is affected by a number of diseases. It is estimated that the loss in cane yield caused by disease ranges from 15 to 20%. Diseases in sugarcane are caused by various pathogens, namely fungi, bacteria, viruses, and phytoplasmas. Among fungal diseases, red rot, smut, wilt, and set rot are significant. Besides these, diseases like yellow leaf disease, ratoon stunting disease, grassy shoot disease, and leaf spot disease have the potential to cause considerable damage depending on the season and variety. Red rot, caused by Coletotrichum falciparum, is the most dreaded disease in sugarcane, being responsible for the elimination of several important sugarcane varieties like CO419, CO997, COJ64, CO1148, CO6304, CO7805, COC671, COC90063, and COC92061 in the past. The external symptoms of red rot appear on the third or fourth leaf, which shows yellowing and drying from margin to midrib. At an advanced stage of the disease, the entire top, including the crown, dries and dies. When the affected stalks are cut open longitudinally, the internal tissues show reddish, discolored patches or lesions interspersed with white horizontal patches. As the disease progresses, the internal tissues become dry and dark in color, resulting in longitudinal pith cavities filled with fungal mycelia. Red rot is primarily caused through infected sets. The secondary spread of the disease occurs during the highly humid monsoon period. Sporulating conidia at the nodal portion of the susceptible canes are carried through rain splash and irrigation water to the adjoining areas, resulting in secondary spread. Fresh surviving debris of red rot inoculum also serves as a source for further spread of the disease. As a control measure, red rot resistant varieties such as CO7704, CO86010, CO86011, CO86249, CO93009, CO94008, COG93076, COSI 95071 and COV 92102 should be grown in the tropical zone. In case red rot resistant varieties are not available, the disease can be managed by following an integrated approach. Sets should be selected from a red rot free area in order to eliminate the primary source of infection. The movement of red rot infected canes from the red rot prone area to the red rot free area should be prevented through domestic quarantine legislation. If primary infection is noticed, then the infected stalks should be destroyed immediately. A plant crop infected by red rot should not be ratoon. Also, the red rot infected plant crop should be harvested on a priority basis in order to prevent secondary spread. Red rot infected sugarcane fields should be rotated with paddy crop to destroy surviving debris-borne inoculum in the field. Smut is another important fungal disease causing loss in quality and reduced yield of sugarcane. It is caused by the fungus Sporicorium cytaminium and is widely prevalent in parts of Karnataka, Andhra Pradesh, and Maharashtra. The characteristic symptom is the production of a long whip-like structure from the terminal bud of the stalk, which is black in color and covered by a thin silvery membrane. This silvery membrane ruptures, releasing millions of reproductive spores of smut fungus, which are present in the form of a powdery mass. The affected stalks are usually stunted. Often, infected clumps produce certain symptoms well before whip production, including stunting, profuse sprouting of lateral shoots, reduction in internodal length, and formation of narrow erect leaves. The disease is more severe in ratoons and more pronounced in dry weather. The smut is primarily transmitted through infected sets. Secondary spread occurs through wind-borne teleospores of the smut fungus. The disease can be easily managed by proper seed selection, roguing of smut-infected clumps, and avoiding ratoons of plant crop with moderate smut infection. In the field, systemic fungicide like propiconazole should be sprayed over the young crop after germination. Sets from smut-affected fields should be treated in hot water at 50 degrees centigrade for 1 hour or 52 degrees centigrade for half an hour along with systemic fungicide to eliminate the set-borne infection. The fungicide hot water treated sets should be raised as part of a healthy seed nursery program and distributed to farmers. Most of the currently released varieties possess resistance to the disease. Wilt of sugarcane is caused by a weak, soil-borne fungal pathogen, Fusarium sacchari. The affected plant appears wilted and conspicuously stunted. The crown leaves turn yellow, lose turgor, and eventually wither. On splitting open the canes at the early stage of infection, diffused reddish-brown patches can be seen. Later, canes become light and hollow and shrink. The disease reduces germination in infected sets, causing drying up of shoots and subsequently results in wilting of the stalks. In severe cases, wilt causes a total loss of the cane field. The wilt syndrome is predisposed by different biotic and abiotic stress factors. Abiotic factors like drought, waterlogging and drought followed by waterlogging weaken the root system and predispose the plant to wilt infection. Subterranean soil pests like white grub, root borer and nematodes. Insect pests like mealybug and scale insect and fungal pathogens like root rot weaken the plant and root system, paving the way for wilt infection. Elimination of these biotic and abiotic stress factors reduce wilt incidence. Wilt can be effectively managed by using healthy sets, crop rotation with paddy, and by application of organic manure in order to increase the antagonistic flora that can suppress the wilt pathogen present in the soil. Grassy shoot disease is a phytoplasma disease widely prevalent in all parts of India. The incidence is particularly high in ratoons and old varieties. Profuse tillering is a typical symptom, with narrow, chlorotic leaves that give it a grass-like appearance, thereby giving it its name. Varying degrees of chlorophyll loss is observed. Very few tillers form canes which are thin and produce white shoots from the side buds. The primary transmission of grassy shoot disease takes place through diseased sets. The disease can cause very heavy yield loss, particularly when the planting material is obtained from infected sources. Maximum yield losses in ratoon occur in crops in which primary infection appears early in the plant crop. Grassy shoot disease can be eliminated from infected sets by treating the sets with aerated steam therapy at 50 degrees centigrade for 1 hour. Roguing and eradication of grassy shoot disease clumps are very helpful in reducing incidence of the disease. Plant crop with high level of grassy shoot disease should not be ratooned. Ratoon stunting disease is caused by the bacterium Lawsonia xyli, subspecies xyli. This disease is considered the most important cause for sugarcane varietal degeneration. Diseased clumps are stunted in growth, thin-stalked with short internodes and have pale yellowish foliage. The internodes taper rapidly towards the top. When split open longitudinally, pinhead-like orange-colored dots can be observed on the nodal region. This symptom is typical of ratoon stunting disease. The disease causes progressive decline in germination and yield. Expression of symptoms is greater under adverse conditions like waterlogging and drought. The disease primarily spreads through infected sets. It also spreads through harvesting implements contaminated with the juice of deceased canes. The ratoon crop suffers more from ratoon stunting disease than the plant crop. To control this disease, the cane cutting knives should be disinfected with 1% Lysol. Hot water treatment at 50 degrees centigrade for 2 hours eliminates set-bone infection of ratoon stunting disease. The 3-tier nursery program is also an essential step in containing ratoon stunting disease and in prolonging the commercial life of varieties. Set rot or pineapple disease is common in germinating sets. It is caused by the fungal pathogen Ceratocystis paradoxa. Typical symptoms of this disease are detected in sets after 2 to 3 weeks of planting. The pathogen enters mainly through cut ends and proliferates rapidly in the parenchymatous tissues of the internode. The affected tissues first develop a reddish color, which turns to brownish-black in the later stages. Cavities are formed inside the severely affected internodes. The presence of the fungus inside the set prevents its rooting, and in most cases, sets decay before the bud sprouts. Occasionally, the disease occurs in standing crop due to the entry of pathogens through the stalk that has been damaged by borers, rats, or through injury. During the early stage of rotting, the fruity odor of rotten pineapple is emitted due to production of ethyl acetate. Set treatment with systemic fungicide, namely carbendazole, at 0.05% concentration is found effective in controlling the set rot infection. Avoiding deep planting during monsoon season reduces chances of disease occurrences in the field. Providing adequate drainage in the field also helps avoid the disease. Maintaining good field hygiene is also important. Integrated management of the sugarcane diseases can best be achieved through the use of disease-free seed sets, suitable cultural practices, and heat therapy with the 3-tier seed nursery program. Yellow leaf disease caused by the sugarcane yellow leaf virus and sugarcane yellow phytoplasma is a recently identified disease in India. It occurs in epidemic form in most of the cultivated varieties in the tropical region. The disease symptoms appear as yellowing of the midrib in the leaves, 3 to 5 in the crown during the 6th to 8th month of the crop. Subsequently, the midrib yellowing spreads to adjoining leaf lamina as a band throughout the leaf from the midrib. Increased severity results in drying of the leaf along the midrib from the tip. However, the disease does not cause any marginal drying. The disease is transmitted through infected sets, and secondary transmission takes place through the sugarcane aphid Melanophis saccharide. The disease expression is influenced by various biotic and abiotic factors. Increased severity of the disease leads to restriction in internode elongation, causing the leaves to appear bunched up. Overall, the severity of the disease drastically affects plant growth and cane yield. The disease can be managed effectively by following a healthy seed nursery program. Meristem culture, combined with molecular diagnosis of the virus, is recommended to eliminate the virus from sugarcane and to develop disease-free nurseries.